In this video, we'll discuss about cryopreservation. Cryopreservation is a process that preserves cells and tissue. So, in this method, we can preserve cells and tissues at a very low temperature to save them for later use. So, in this cryopreservation method, we can preserve cells and tissue at a low temperature to save them for later use. So this method is also known as cryobanking. How it work? How that cryopreservation method will work? Cooling the cell to very low temperature between -80 to -196 degrees Celsius. So here we can preserve cell between -80 to -196 degrees Celsius to suspend their metabolism. This process protects the cell organelles, tissue and other biological component. So this process protects the cell organelles, tissue and other biological component. This cryopreservation method having two types. One is slow freezing. Second one is vitrification. So slow freezing and vitrification method. What is slow freezing method? Slow freezing is a cryopreservation technique that gradually cools cell to preserve them for long period of time. Used for cryopreservating. By using this method, oocytes embryo for in vitro fertilization IVF treatment that oocytes and embryo we can preserve by using this method. Then vitrification method involves rapidly cooling liquid water below its freezing. So this method rapidly cooling liquid water below its freezing point to preserve cells, tissue and other biological system without ice formation. So it will store without ice formation. This method name is called as vitrification method. This method also used in fertility preservation like embryo, oocytes, ovarian tissue and stem cell culture, 2D, 3D cell, stem cells and organ preservation we are using. Then what are the factors that affected the process of cryopreservation? The type of cell being preserved, the rate at cooled and the temperature it's frozen to can also affected that outcome. So what are the factors affect? The rate at which it cooled and the temperature it's frozen to can all affect the outcome. Then cryo injury, damage that occur to cell during cryopreservation. Sometimes damage also will occur. That one name is called as cryo injury. Caused by factors such as incorrect cooling rate, extreme temperature and cell being preserved. So cryo injury during preservation time it will cause us injury. So factors incorrect cooling rate, extreme temperature and cell being preserved. Then last one is application used in a variety of scientific investigation and it has many clinical application. Scientific investigation and clinical application we are using this cryopreservation. So cryopreservation. Thanks for watching.